Hi friends, welcome to Madhu's teaching. In this video, I want to tell about what are the two types of vectors. One is cloning vector and another one is expression vector. Cloning vector to generate and maintain multiple copies of our DNA fragment or gene of interest. Gene of interest means which gene we want to insert into our host organism, that is called gene of interest. We use cloning vector to maintain multiple copies of our DNA in the host organism. Next, expression vector to produce more protein from our gene of interest. We use expression vector to produce more protein from our gene of interest. Means in expression vector our gene will be expressed. Means conversion of DNA to mRNA, mRNA to protein. We use cloning vector to maintain multiple copies of our DNA fragment. We use expression vector to produce more protein from our DNA fragment. Next, cloning vector contains ORI, means origin of replication. Origin of replication means where the initiation of replication is known as origin of replication. Next, selectable markers. Selectable markers are antibiotic resistant genes. So cloning vector should definitely contain the selectable markers. Selectable markers are helps to identify our gene of interest whether it inserted into our host organism or not. Next, MCS. MCS means multiple cloning sites. Multiple cloning sites means six nucleotides are linked together to form a polylinker, that is known as multiple cloning sites. Multiple cloning sites is a site where our gene of interest should be inserted. So cloning vector should definitely contain the multiple cloning sites. Next, expression vector should contain origin of replication, selectable markers, MCS, and it should contain extra elements that are regulatory elements. Promoter. Promoter region is a site where the RNA polymerase will binds. When the RNA polymerase will binds to the promoter region, from there only gene expression will be started. When the RNA polymerase will binds to the promoter region, then RNA polymerase will synthesize the mRNA. Next, mRNA will be converted into protein. So promoter is very important to expression vector. Next, enhancer. Enhancer enhance the function of RNA polymerase. Next, transcription termination signal. It terminates the transcription process. Next, reporter genes. Reporter genes are to monitor the gene expression. Examples. Most common example of cloning vector is PBR322. Next expression vector example is PET vectors, that is PET28A and PET30A.